Good morning, St. Louis, and good afternoon, Leverkusen. Here, I'm very glad to give this presentation about recent finding starting from marine extreme environment in order to find out novel bioactive molecules. Just few words about where I came from. I belong to the Stazione Zoologica Anton Dorn, which is a public research organization that was funded by Anton Dorn, a German scientist, in 1872, with the mission to promote basic research. So the idea, the basic idea of Dorn was to establish several stations, marine stations around Italy. And he established the first in Naples, the second in Ischia, and the third in Messina, in Sicily. And it was a very great idea, I think, because the idea should be to have a free space, a free lab in which all the scientists can do research on biodiversity, ecology, stressors in ecology, and try to understand more about the marine environment. Now the Stazione Zoologica is a very modern organization. Has a very modern organization. We are around 260 people as a permanent staff, and more or less the same as a temporary staff. We are divided in 5 different departments and with several premises around the peninsula, because we have premises in Ischia, Portici with the Turtle Point, Bagnoli with the marine farm, Messina, Palermo and Milazzo, which are mostly focused on fish and fish management, Amendolara for the monitoring of the coast, Fano and Genoa on marine protected area. The newborn Department of Sustainable Marine Biotechnology is located directly on the sea. This is a very nice picture in which we can understand here with the first arrow the location of the department that has the vision to stay on the sea and very close to the research vessel Victoria that is located here in the harbor, in order to have a direct access to the marine organism, in order to understand if they can possess some compounds that could be useful for biotechnological applications. So just a few words about me, because I have been a researcher at the National Research Council for 20 years, and after I joined the research, the Stazione Zoologica since 2019, where I became the director of the department. Just a few words about the department, which is a unique department in Italy and one of the rare example department entirely focused on marine biotechnology. We have the mission, of course, of the department is to explore marine biodiversity in order to sustainably exploit marine organisms, in order to find out compounds useful for some industrial application like pharmaceutical, nutraceutical, and cosmeceutical industries. But also we have environmental biotechnology, aims to isolate novel bacteria and microalgae able to useful for the bioremediation of environment, and also on the biomaterial biotechnology. We are very active in many different sectors, at least also in the fish waste valorization. All these research line falls in the blue growth and blue economy. Smart and sustainable and inclusive economic and job growth linked to the ocean. We have several sectors that are important for the blue growth and the blue economy, like the preservation of living and non-living marine source, renewable energy derived from the sea, port activities, naval sectors from construction to maritime transport, coastal tourism, fishing, aquaculture, and marine natural products. This is really important that here in Naples, from the department, we can of course be connected and all the research activity will fall in order to develop and exploit the European Green Deal. Apart from the main sectors, we have also other subtopics which are related to the application of synthetic biology in order to introduce some cutting-edge technologies, and also we have other subtopics related to the drug discovery, to the biomonitoring and bioremediation of pollutant and the waste valorization. Just want to show very brief how we are organized. We have inside the department 3 different functional areas, and a functional area related to the pharmaceuticals, cosmeceuticals, and nutraceuticals. Coming back to the main objective of this talk, we are focused on the exploitation of marine environment. Why marine environment? Because it possesses a high biological diversity, a broad range of different habitats, complex interaction, competition, defense, and communication are really different from the terrestrial one. And in particular, microorganisms have adapted and have developed specific adaptation to extreme environments like deep sea, like in Arctic and in Antarctic, high pressure, salinity, extreme temperature, and darkness. And in particular, the adaptation include, of course, the expression of secondary metabolites that may have potential novel biological activities. And the bioprospecting of this natural product can lead to the isolation and characterization of novel compounds. In fact, exploring extreme and unexplored environments may lead to the discovery of new class of natural products. In this ambit, psychophilic bacteria represent a very good source of unexplored biodiversity, since they are able to survive and they are adopted survival strategies in order to thrive and face adverse environmental conditions, achieving competitive advantage. And also they may produce interesting compounds with some specific activity that are really important for us. It is really important to understand that ocean, which covers 70% of the surface Earth, we have explored at least only 5%. So we have hidden treasure to be discovered and to be exploited. This is one of the examples of one of the best compounds that have been exploited from marine source. Herein, I just want to give you some update about where we collected our samples. We collected in many different parts of the Earth, starting from Svalbard, Barents Sea, coming through some specific area, and also extreme environment like in Ischia Island, in the CO2 vents, or in the river of at the end of Sarno River. It is one of the most contaminated and polluted river in the world, in Europe, and also in the Bagnoli Bay. We were on the Tibetan glaciers in order to collect some sediment samples from glaciers, in the Canary Island, on Panama, and also in the South Shetland Trench, in the Deception Island, and of course in Antarctica, over many expeditions that we performed. In particular, what I want to highlight today is some specific strategy that bacteria can adopt in order to survive in this harsh condition. So Antarctic and Arctic regions seems to be very similar, but they are more dissimilar than similar. In fact, first of all, Antarctica is a land surrounded by the water, while Arctic is water surrounded by the land. And while Antarctica is completely isolated from the rest of the world, since the presence of circumpolar current, Arctic pole is completely surrounded by land and also influenced by the anthropogenic activities. So for this reason, Antarctica is a natural laboratory in order to understand how microorganisms and organisms in general have adopted their strategy in order to cope with these harsh conditions, and also how organisms from the polar habitat are affected by the climate change. I just want to talk a very little bit about the evolution in the cold, since the Antarctic biotopes offer this kind of opportunity. And in particular, cold-adapted bacteria are generally acknowledged to achieve their physiological and ecological success in cold environments, such as results in specific structure and functional properties in their genomes. Just a few examples. For example, the membranes, they should be a little bit more fluid at low temperature. For this reason, the presence of unsaturated fatty acid are much more abundant than the mesophilic bacteria. The presence of desaturases, lipid desaturases are really important. Freeze protection. How to reduce the freezing point of the cytoplasm through the presence of osmolytes, so compatible solids like betaine or trimethylamine oxide, antifreeze protein, ice-binding protein, which are able to reduce the freezing point of the cytoplasm. Cold shock and acclimatization response. We are able to highlight and detect some hot heat shock proteins, compatible solute again, RNA binding proteins which preserve which increase the cellular response to lowering of temperature. Protection of reactive oxygen species. And so we highlight the presence of much more catalyzes, peroxidases, superoxidodismusases. Protein and enzymes. We recognize some specific feature in proteins and enzymes by the reduction of disulfide bond, by the reduction in number of proline, in order to make more fluid and in order to allow them the catalysis of the reactions at very low temperature. So proteins are more flexible at low temperature. And of course, a great genomic plasticity thanks to the presence of transposases and prophages. These are more or less the specific adaptation of psychrophilic bacteria that we can recognize on the opposite when we study thermophilic bacteria, in which we can find a different adaptation which may allow the catalysis at very high temperature, around from 60 to 80 degrees centigrade. So the identification of bioactive compounds from marine organisms allowed us to identify many different compounds that could be useful in many different sectors, like agriculture, medicine, pharmaceutical, cosmetic industry, and aquaculture. So we are in a field in which we can obtain different compounds that could be applied in different sectors. Again, we are working on a surface of the ocean, which represents 70% of the total surface of the Earth, that contains around 10 to 9 bacteria and archaea, so which is an enormous quantity of bacteria, fungi, and microalgae. Very little is known about this biodiversity if you consider that around 5% of the total biodiversity of microorganisms have been discovered so far. So we are in a presence of huge biodiversity to be discovered. The success of this bacteria in every environment is due to also to the flexible metabolism that bacteria possess in order to respond to the ecological stress in a very short time. Food shortage, the complex situation of competition, low temperature, high pH, low pH, pressure, they have the capacity to react very, very easily in order to achieve the competitive advantage. So, coming back to the science, what we did in the past is to establish a traditional biodiscovery pipeline, which starts from the sampling, the collection of sediment samples, go through the isolation of the microorganisms, strains identification, cultivation, extraction of compounds, bioguided assay fractionation through screening platform, and the purification by HPLC and identification of the molecules. Just a few words about this traditional biodiscovery pipeline, which starts with the traditional approach with the isolation of sediment samples and the serial dilution in order to get discrete number of colony that can be isolated, and the microorganism identification can be done through the extraction and the amplification of 16S DNA genes in order to find out and check the specific strains. Followed by the screening, and we set up here during last year different screening, liquid screening for the presence of anti-biofilm compounds, antioxidant, anti-cancer, and antimicrobial assay, and plate screening for the hemolysis assay, siderophores, biosurfactant assay by using CTAB. So, after of course, our extra are tested for a different panel of bioassays. The purification is performed by following the activity. So the screening highlight the presence of putative positive microorganisms that are selected again and fractionated in a different way by using different organic solvents, and the activity is evaluated with antimicrobial assay, with the MAC assay and NBC assay, in order to define the inhibition of the growth of the strains of Staphylococcus aureus, for example, and after the purification of the pigs is allowed by the use of HPLC. The molecule identification is done through the mass spectrometry and NMR. So, but now we are optimizing the biodiscovery pipeline, going to take samples from extreme and unexplored environment, try to improve the microbial cultivability of the uncultivable strains, use the metagenomics and the genomics approach in order to have the direct genomic analysis, apply the OSMAC approach in order to maximize the production of secondary metabolites, improve the organic solvent extraction in order to reduce the amount of organic solvents used, high throughput screening for the isolation of putative strains, and the improvement of metabolomics analysis in order to understand the metabolic pathway for the compounds isolated. So today I just want to talk about a recent finding obtained in our lab regarding biosurfactants. Biosurfactants are amphipathic molecules produced by many microorganisms that are composed by hydrophilic heads with dinidophobic tails. Marine organisms are able to produce many different kinds of biosurfactants coming from lipoproteins, glycolipids, polypeptides, and fatty acids, and other lipids. And they are very important lipids, they are very important molecules. Why we study biosurfactants? Because they typically exhibit higher activity at lower concentration compared to many synthetic biosurfactants, showing a very low critical micelle concentration, low toxicity, high biodegradability, and tolerance to extreme conditions such as temperature values, extreme pH, and high salinity. So biosurfactants are really important for some industrial sectors because they are very considered very attractive for a commercial point of view because of their use in agriculture, such as a biocontrol agent thanks to their insecticide activity, or the application in bioremediation, assisting hydrocarbon pollutant biodegradation and metal sequestring, the chemical, food process, food additives, cosmetics, pharmaceutical industries are very interested in the production of biosurfactants. So, again, a good strategy, of course, is bioprospecting of marine environment at the deepest, the coldest, and the darkest part of the heart. Herein, I just want to give you some results about what we did in the last years with the discovery of new biosurfactants from marine environment, but in particular from the Antarctic and Arctic strains. We applied we set up the biodiscovery process, but and so therefore we collect environmental samples, we isolate marine bacteria, and after we go through two different approaches: the classical approach, which give us some results, and the modern approach. And just want to highlight these two different approaches. We performed several expeditions, several field campaigns. In particular, just want to talk to you about the sampling campaign that we performed in the Barents Sea on board of Helmer Hansen vessel in collaboration with the Norwegian College of Fishery Science in Tromsø, Norway, and we collected in 9 different sites in the Barents Sea close to oil rigs. And this expedition was really good because we had the chance to have the chance to collect sediment samples in a very remote area of the planet, very close to the Svalbard, and at very high latitude. And also we participate in the expedition, the PharmaDeep expedition, coordinated by the University of Aberdeen in the Eurofleet program, in which we collected sediment samples, animals from South Shetland Trench, at least around 6,000 meters of depth. And it was really a harsh condition, first of all, because it was a cruise performed during Christmas time, and so it was a great chance to have access to these sediments, animal that we never never exploited before. So after the collection of these sediments, we isolate bacteria that were sediments are mixed with saline solution and in a different condition, and we were able to isolate around 800 microorganisms. And to the classical approach, we analyze these bacteria in order to understand if they are able to produce biosurfactant through the 3 different assays. The first one is the CTAB blue agar assay that reveals the presence of an anionic biosurfactants that form blue precipitate with the CTAB and methylene blue. The emulsification index, which highlights the ability of a biosurfactant to stabilize the emulsion, and the oil sprayed assay. Herein, maybe you can see in this short movie, this is the ability of a biosurfactant to clarify an oil layer on the water. Also, apart from the 800 isolates from the Deception Island, we also screened some bacteria, some 24 bacteria that we isolated from Baia Terranova, that is the Antarctic Italian base, and we isolated these strains from the water column and the sediments very close to the Antarctic seawater. After the screening of these bacteria, we cultivated them in the liquid media, and we extract all the compounds by using ethyl acetate, and we evaporated all the solvents by using the rotovapor. Herein we highlighted some results related to 3 strains, M15, M38, and T28, which results the best producers of biosurfactant. So the strains, of course, are really important. The cultivated, of course, in only 1 conditions. After that, the LCMS analysis revealed a very complex chromatogram with many peaks, and we discovered that these peaks revealed the presence of complex mixture of rhamnolipids with few new congeners. Rhamnolipids are a class of glycolipids formed by sugar moieties represented by rhamnose, and the lipidic part composed by 2 hydroxy fatty acids. I just want to highlight this is what we did by using the classical approach. Now I just want to talk to you about the second strategies that implies the use of most cutting-edge technologies, most updated technology, the omics approach, and we focused our attention on a strain Rhodococcus I2R, which belong to actinobacteria, and this genus is very industrially relevant, hosts huge metabolic diversity, highly producer of bioactive compounds, and widespread in different habitats, including extremes 1. What we did is to sequence the genome and to perform a genome mining by anti-smash platform, but also what we did is was to apply OSMAC approach, which means 1 strains many active compounds into the 22 different grow conditions. So the same bacteria was grown in 22 different conditions, varying the culture media to enhance the production of metabolites. And the extraction of by ethyl acetate of these 22 different conditions allowed us to perform the metabolomics plus functional screening. This is some of the results that we obtained. So for the metabolomic analysis, Each extract was submitted to a mass spectrometry analysis, and all these spectra were analyzed and the molecular network was performed. Here in circle represent molecules, and the sides represents the compounds of relative concentration in the extract. Then we see here the color code. The colors are representative of each medium and tell you in which medium they are produced. So as you can see, there was a family here in the square in which this is the only compounds that has been produced by using on this SV2 medium. So this is a very complex analysis, and this is a really important analysis in which this is the unique compounds that is produced in these conditions. And this approach may allow us to discover these succinyl saccharide esters that is a glycolipids formed by 3 of 2 glucose units that are linked to fatty acid. And this also contains succinyl acid, and the presence of succinyl acid allowed us to understand that it is a sort of biosurfactant because it is active to the blue agar assay. Very often it happens that since we have a large number of bacteria that we have to screen, we have to mix these two approaches, and so the metabolites production are performed in the deep well in order to maximize, to have 96 different conditions in order to have to maximize the production of the metabolites that, of course, after are detected by the CTAB agar assay and oil spreading assay. In the mixed through the mixed approach, we were able to screen in 2 weeks around 300 strains, screened in 4 different conditions, which highlighted the presence of 30 positive strains, and that were, of course, sequenced in their genomes. And the metabolomic analysis allowed us to identify compounds that are produced in only 1 condition. That is really, really important for us. So what we discovered from the M15 rhamnolipids from Antarctic bacteria. These rhamnolipids possess a very interesting activity as antimicrobials, when tested towards Staphylococcus aureus reference and clinic isolate with encouraging results. Then a time-killing curve was performing, showing an MIC value that corresponds to NBC, killing bacteria in 5 hours. Also, what we realized is that rhamnolipids possess also a very important antiviral activity that we tested after the COVID outbreaks. And these are really important for us, but I will not go into details about this. What we are studying now is the biotechnological application of our Antarctic rhamnolipids, because we would like to apply these rhamnolipids in food dressing. Chronic foods affect around 20 million people worldwide annually, and often are associated with microbial infection. And one of the prevalent infection agent is Staphylococcus aureus. Since our rhamnolipids are particularly active towards Staphylococcus aureus, We would like to apply our rhamnolipids to a wound dressing, stop bleeding, absorb the excess of fluids, try to understand if it is possible to create a barrier from the external environment, and an antimicrobial treatment with treating the dressing with our rhamnolipids. Of course, the bottleneck of this research is the yield that we can obtain from the natural sources. For this reason, what we did, and maybe we can discuss later about that. We are heterologous express the gene responsible for the production of monorhamnolipids and dirhamnolipids isolated from M15, because it is really important to have some industrial application, to have a recombinant production of these compounds that could be really important for some industrial sectors. So at the end of this very nice talk, I think that biosurfactants are valuable surface active molecules that possess many bioactivities, but it relies from, we start from the bioprospecting of marine environments, in particular the bioprospecting from unexplored and extreme environments is a proven strategy for the discovery of novel compounds. We trust in biosurfactant research because we are strongly convinced that natural biosurfactant will take the place of the surfactant, the chemical one, which are up to now used for industrial application. Because the heterologous expression, of course, is a necessary step to overcome the bottleneck for the production and differentiate, of course, the production. This is all for me, and thank you for your attention.
Thanks very much, Donatella, for this really great talk. It was interesting to see how to really exploit these wonderful organisms, not just to only see them, to study them, but also make use of their fascinating features. And I'm really especially surprised to see so many glycans which play a role in your research. And I suppose that we will have quite some questions from the auditory also with regard to those compounds. So the floor is open. Here you are.
Thanks a lot for this wonderful talk. This was really very fascinating. Michael Beck talking from Monheim. I have not a question on the compounds, sorry, but on the proteins actually, because I never thought about that, but especially on the cold environments, the proteins must be utterly, the enzymes must be really effective, right? Energy effective. So is there anything going on to exploit such proteins in the context of industrial biotransformation for a more sustainable production?
I'm sorry, proteins?
Yes, the enzymes. So the enzymes for these.
So in the past many years, esterase and lipase isolated from psychobilic bacteria. And of course, they have a huge application in the laundry because, of course, with the energy increase the cost, it is really important to obtain soap and additives that could have their action at very low temperature. About the enzyme in particular, the esterases and lipases, we found from Antarctic bacteria that we isolated in the past some esterases that are able to catalyze their reaction at 10-15. It is really energy saving for industrial application. One of these, one example is the lipase from Candida antarctica, that is a marine fungi, an Antarctic marine fungi that is applied in the industry. Another example is the antifreeze protein that Unilever for the first time used it in ice cream, because the antifreeze proteins are proteins which reduce the freezing point, and they found application as a food additives in the ice cream productions, in order to have the soft cream and in order to increase also the taste of the ice cream. So they found a very huge application in the industrial sectors. Another one could be the chaperone, because, for example, for heterologous expression of proteins, very often inclusion bodies are prevented by the use of molecular chaperones isolated from Antarctic bacteria, which means that they are able to express heterologous express proteins at very low temperature, using the E. coli temperature from 37 to 15 degrees centigrade.
Many thanks. And just a short question in St. Louis, perhaps?
Yes. Hello, here from St. Louis. Thanks for your talk. I wonder, all these applications you have found in this biodiversity from Antarctica, how you manage the regulatory approvals or Nagoya treatments for applying, take this from biodiversity to the industry?
Yes, so Antarctica is out from Nagoya, but we have Antarctic treat. It is a different approach, because from the Antarctic seawater you can collect biodiversity, you can study, but not commercialize. So we are talking about possible applications, but now we never applied anything right now. We know that other psychophilic bacteria, other esterases, some enzymes are isolated from not from Antarctica, but from international seawater. So which means that they are out of Nagoya, and their application is easy. What we can do in Antarctic is to collect for scientific purpose, but we cannot patent anything, because which means that we cannot exploit commercially for this. What we can do is different application, different patenting for obtaining commercial exploitation.
Thank you so much.